This video lecture will cover Chapter 8. It's the second half of Chapter 8, so Part 2. We'll be talking about cell-to-cell nervous communication, synapses, and neurotransmitters. So first, let's define a synapse. If we have a neuron, and we went over the different parts of the neuron last time. So we have its soma, its dendrites, the axon, and then we come to the axon terminals. And on our axon terminals, we would then have them terminating on another axon. This space between the synaptic terminal and the dendrites of the next axon or the next neuron is what we would call the synapse. Okay, we have two types of synapses. We can have an electrical synapse in which we are just changing membrane potential between the axon terminal and the membrane of the next neuron. Or we can have a chemical synapse in which we're releasing an actual chemical. It binds to a receptor and that initiates a response. This figure is taken from your book, and it's a great figure because it outlines all of the steps in axon neurotransmitter release. So when we initiate our action potential at the axon hillock, we start a depolarization chain reaction that is propagated down the axon to the axon terminal. So as this depolarization moves along the membrane and it reaches the axon terminal, this depolarization causes calcium channels to open. So we have gated calcium channels, and what occurs is we have an influx of calcium into the cell. As this calcium concentration, the intracellular calcium concentration increases, that triggers exocytosis of synaptic vesicles. And a synaptic vesicle is a membrane-bound vesicle. Inside are packaged neurotransmitters. And so these membrane vesicles, they'll fuse to the membrane and then release those neurotransmitters into this area between the axon terminal and the next cell. We call that the synaptic cleft. So neurotransmitters get released into the synaptic cleft. Those neurotransmitters then bind if we have the appropriate receptor protein on that membrane. They will bind, and that triggers a cellular response. So these steps of depolarization, calcium gates being opened, calcium intracellular concentrations increase, that triggers exocytosis, and neurotransmitters get released in the synaptic cleft, is what occurs at the end of each action potential. Now, typically, when these synaptic vesicles fuse, they just become part of the membrane. Okay, new vesicles will be made in the soma of the cell and be brought down the axon with neurotransmitters and be set in there. That's the most common fate of these vesicles. There are other pathways. One is the kiss and run pathway, in which the vesicle will actually fuse with the membrane at what we call a fusion pore. So previously you see we have these docking proteins that they kind of bind to, but then this will be incorporated into the membrane. When we have a fusion pore, that holds the vesicle in place. Neurotransmitters can pass through that channel, but the vesicle can then actually pull back from the fusion pore and remain intact. The advantage of this is that the cell doesn't have to continually make new synaptic vesicles. Okay, we call that the kiss and run pathway. So what happens when we release neurotransmitters? We talked about one. There's really one of three fates. The first one that we already kind of talked about was that it binds. Okay, but if it doesn't bind to a receptor, what happens to it? Well, Firstly, it can either be returned into the axon terminal for reuse. There's membrane proteins that can actually pump it back in, and then they can be repackaged into a synaptic vesicle. Or sometimes, which is what we can also see happen, is that they will be pumped in to a glial cell. So remember, glial cells are accessory neuronal cells. And so it'll be moved into the glial cell, and there it can be either recycled, broken down, what have you, inside the glial cell. That's the first fate, either back into the axon or into a glial cell to be reused. The second fate is there are enzymes, so proteins that function as an enzyme that are embedded on the membrane of the postsynaptic cell. And I didn't really talk about that terminology. Typically when we have a synapse, right, we refer to the axon terminal as the presynaptic cell, and the cell that's on the other side of the synapse is the postsynaptic cell. So on the postsynaptic cell, there are proteins that serve as enzymes that can break down the neurotransmitters, that break it down and remove it from the synaptic cleft. And then the third thing that can happen is that we can just have diffusion, in which the neurotransmitters diffuse out of the synaptic cleft. Maybe they move into a blood vessel, maybe they move into other intracellular spaces, but they leave that synaptic cleft. Why do we have these three pathways? Well, again, these are signal molecules, right? And so we don't want them just sitting around here forever, signaling the cell to function. So from a physiological standpoint, we have a very quick release in which a small amount of neurotransmitter gets released into the synaptic cleft, and it binds, initiates a response, but then it's also being removed so that we can eliminate that response and have a fast signal molecule. How does our body regulate how much neurotransmitter is released? So we have an action potential, and then we initiate those calcium. The depolarization causes our calcium gates to open. Calcium comes into the cell. That triggers exocytosis of the synaptic vesicles, and we release neurotransmitters. But how does the body regulate how much gets released? To answer that, we have to look at the frequency of the frequency of the action potentials and how that correlates with neurotransmitter release. And what we see is when we have a suprathreshold graded potential that then triggers an action potential, for each action potential there's a small amount of neurotransmitters. In this case, when we have an even larger graded potential, right, that larger graded potential may last through the refractory period of the first action potential, and so we get even more action potentials because after the absolute and relative refractory period, it's still above threshold, so we initiate another action potential. So as the frequency of the action potentials becomes more, so we have more action potentials, we get more neurotransmitters being released. So you can think about it like an action potential being a tap of your finger, right? For each tap, X amount of neurotransmitters is going to be released. So if you tap more, right, you're going to release more neurotransmitters out. I like to think of it kind of like a video game, like old school video games where if you push the B button, that fires your gun, right? And you get one shot. But if you want to like fire a ton of shots, it's not like you can just necessarily in some of the old school games hold down the button. That doesn't work. You have to end up like tapping as many times as possible. And so the faster you tap, the more you're going to be able to fire a gun, or the more you're going to be able to kick, or whatever, in whatever game we're talking about. That's what's going on with the neurotransmitter. So frequency of action potential equals more neurotransmitters being released. And again, remember, all action potentials have the same amplitude or the same strength. So it's not like you can have a stronger action potential. No, you can have a stronger graded potential. The stronger the graded potential, the more action potentials you'll have. that brings us to this concept of divergence and convergence neural pathways. So as we start to have these synapses, what we see is that we can have different pathways. We can have a pathway in which the axon terminals trigger more and more cells, and we kind of have this pyramid, and we can have what's called a convergent pathway, in which we have lots of neurons that are then converging. So they're providing influence to a smaller number of postsynaptic neurons, and our triangle then would appear this way, right? What's the benefits of these two types of pathways? When a divergent, we start with a single stimulus, single area, and then we send it out to lots. We get a lot of divergent pathways when we are talking about integrating centers in the brain and then sending signals out for a systemic response. Convergent pathway, though, this might be more of, say, like an afferent signal, where maybe we're getting lots of different signals from all over the body, and it's being brought in to a single region in the brain to then be interpreted. So our body uses both divergent and convergent pathways to function and send signals. Another unique aspect of our graded potentials that we didn't necessarily talk about last time, but we need to talk about now, is this concept of what we call summation. So spatial summation and temporal summations are ways in which small sub-threshold graded potentials can actually trigger an action potential. So let's look at spatial. In spatial, what we're saying is that we have multiple neurons that are there for sending an action potential at relatively the exact same time. So this neuron is sending an excitatory action potential. This neuron is sending an excitatory action potential. Both of those membrane potentials, when they're received as a graded potential, so those neurotransmitters bind to receptors, they change the membrane potential. You'll see that both of those are sub-threshold, but because they're being received from two different areas, and relatively at the same time, we can add them. They're additive. So they get added to each other, and they together are strong enough to trigger an action potential, whereas by themselves they would not. So this is spatial summation, coming from two areas but being received at pretty much the same time. Now in temporal summation, we're getting many excitatory potentials from a single neuron, and they're occurring in rapid succession. So we have one graded potential that changes membrane potential, and then let's say it starts to kind of go back down, but then we get another excitatory. So rather than returning all the way to membrane, to the resting membrane potential, it gets additive and adds up again, and we now have a suprathreshold graded potential, and we trigger our all or nothing action potential response. So keep those in mind, that we can add these, and again, on graded potentials, there's not really that like in the action potential, that same absolute and relative refractory period. So that's why it allows for summation to occur. Now let's bring in one other aspect of this, is we can have what we call both excitatory presynaptic potentials, or we can have inhibitory presynaptic potentials. So an excitatory one is going to depolarize the membrane. An inhibitory potential is going to hyperpolarize the membrane. And so not all neurons excite. Some cause hyperpolarization. And these two can be summated and added. So in this case here, we're receiving an excitatory action potential, but another neuron is doing an inhibitory action potential, and they cancel each other out. So maybe this one was initially, strong enough to where it could be summated if we had a second one, but this cancels it and returns us back to resting membrane potential. So you see how the body can utilize these responses to inhibit or excite postsynaptic neurons. Now, I just started covering this, let's put some terms to it. So synaptic inhibition is now where we would stop the release of a neurotransmitter. And there's two ways that can happen. One, we can have what's called presynaptic inhibition. Like the name implies, in presynaptic inhibition, it's going to occur before the synapse. So here's an example. We have an excitatory neuron that depolarizes the membrane, the soma of a postsynaptic neuron, which triggers an action potential. We send that action potential down the axon. As it comes to the presynaptic terminals, because of the network of neurons we have, oftentimes there can be an inhibitory neuron that will release neurotransmitters right on the axon terminal or on that membrane. And if this one is triggered, we can stop this depolarization from triggering the release of neurotransmitters. This would be a presynaptic inhibition. Now what you'll notice is on other axon terminals that don't have that inhibitory neuron, they still release the neurotransmitters and we get a response. So presynaptic inhibition can be a very fine way in which the body can inhibit one selective target. Postsynaptic inhibition is now going to occur on the postsynaptic neuron itself and completely turn it off. So that's kind of what we were talking about on the previous one. Here we have an excitatory neuron. It releases neurotransmitters that then, say, open sodium gates that would cause an excitatory postsynaptic potential. But we could have an additional inhibitory neuron that releases neurotransmitters that open up, let's say, chloride gates. Those chloride gates are going to counteract the EPSP. It's going to be inhibitory postsynaptic potential and cancel out that graded potential so that when we reach the axon hillock, there is no super threshold graded potential. We're below threshold and we don't send a response. So postsynaptic inhibition, all the targets are going to be inhibited equally. So at the end, none of our target tissues have a response because we shut the whole neuron down. That finishes up what I want you to know about synapses and synaptic inhibition, summation. So now let's move on to the neurotransmitters themselves. So neurons release what we call neurocrine signals. These are signal molecules that are utilized by binding to receptors and eliciting a response. And there's three main types. There's what we call neurotransmitters. Neurotransmitters do not, they bind directly to receptors in a synapse. They don't diffuse far away, and they usually open ion channels. We can also have what are called neuromodulators. So a neuromodulator is similar to a neurotransmitter, but generally its mode of action, what it's going to do is it's going to alter the functioning of a membrane protein. Okay, it's not going to necessarily open a gate or it's going to alter some function. And lastly, we can have neurohormones. Neurohormone, again, very similar to a neurotransmitter, except as the name implies, it will be circulated in the blood. So those are our three types of neurotransmitters. When we talk about neurocrine receptors, we have two types of receptors that can be found on our target tissues. We have those that we call ionotropic. So the ionotropic receptors, when we have a neurocrine bound to an ionotropic receptor, it will open an ion gate. Okay, so we have a gated channel, it gets open, and ions will enter into the cell, like sodium or chloride, potassium, calcium, what have you. The other type we have is what we call metabotropic. So in a metabotropic receptor, now when we have a neurocrine bind to it, what it will do is it's coupled with something like a G protein coupled cascading signal molecule. So when we bind to that receptor, let's say we have a cell, here's the receptor. Okay, in the ionotropic, it's a gated channel, so when we bind a neurocrine to it, we bring things through. In a metabotropic, when we bind to this receptor, There's proteins that are associated with it on the inside of the cell. Generally, these are what we call G proteins. These get activated, and it has this cascading response that ultimately releases an intracellular signal. So we have a signal that can change some aspect of functioning. Maybe we turn transcription or translation on in the cell. Maybe we alter some enzyme reactions inside the cell. But nothing actually enters the cell like we do in an ionotropic. Neurotransmitters can be divided into 7 classes, and these 7 classes are dictated by their structure. I expect you to know these 7 classes. So you need to know that we have acetylcholine, amines, amino acids, peptides, purines, gases, and lipids. And let's talk about them each briefly. Some of these are going to be much more important than others, that we'll talk about for the rest of the semester. First of those really important ones is going to be acetylcholine. Acetylcholine is made by taking a choline molecule and adding it to an enzyme called acetyl-CoA. So the acetyl coenzyme A goes together and we get acetylcholine. And acetylcholine is released by neurons that we call cholinergic receptors. Okay, so sorry, cholinergic. Acetyl-CoA gets released by neurons that are called cholinergic. So cholinergic neurons, their neurotransmitters, will be acetylcholine. Acetylcholine then binds to cholinergic receptors. There's 2 types of cholinergic receptors that we will be talking about in depth this semester. The first type is called nicotinic. It's named after an agonist, nicotine, like what's found in cigarettes. Okay, nicotinic receptors are found in our skeletal muscles, autonomic neurons, and in the central nervous system. The other type is called muscarinic. Okay, again, it's named for the agonist muscarine. Okay, muscarinic receptors are found in our smooth and cardiac muscle, and in endocrine exocrine glands, and lastly also in the central nervous system. Another group of neurotransmitters is the amines. So the amines, they're derived from a single amino acid, and something that's unique about the amines is they're only active in the central nervous system. So we'll only see them in central nervous neurons. We won't find them in peripheral neurons outside the body. Or outside, sorry, not outside the body, but outside the central nervous system. Some of these single amino acids include, so we can take tryptophan and it gets produced into serotonin. Histidine can get produced into histamine. And then the big one that we'll probably kind of follow most of is our tyrosine amines. So tyrosine is utilized to make dopamine, norepinephrine, and epinephrine. And while we'll talk about dopamine, really for most of the semester, we'll be focusing on pathways that utilize norepinephrine and epinephrine. The neurons that release norepinephrine are what we call adrenergic. Okay, sometimes we'll also call them noradrenergic. But adrenergic, again, it comes from the compound that was first identified as being an agonist, meaning that it triggers those receptors, which was adrenaline. So adrenaline was first described in England. But we come to find, that adrenaline really is norepinephrine. Okay, that's the actual chemical name for it. So adrenergic neurons secrete norepinephrine. And the receptors, the adrenergic receptors, we have 2 types that we'll talk about. We have alpha adrenergic receptors and beta receptors. And I'm sure some of you have heard the concept of like beta receptors because of the drugs beta blockers that are used for high blood pressure and cardiac issue. We'll talk more about that when we get to the cardiovascular system. Both of these receptors, they're G protein coupled. Okay, so they're metabotropic. We have these second messenger pathways. Okay, the acetylcholine and our amine, so the norepinephrine, are going to be the main neurotransmitters that we'll be talking about for the next little while. But just to kind of cover the other ones, we also have amino acids that can serve as neurotransmitters. There's several amino acids that function this way. Glutamate is the primary excitatory neurotransmitter released in the brain. We have, with that, in the brain, the main inhibitory neurotransmitter is gamma aminobutyric acid, or we refer to it as GABA. So glutamate, primary excitatory neurotransmitter in the brain. GABA, the main inhibitory neurotransmitter in the brain. And then glycine, this one is also inhibitory, but we utilize it in the spinal cord, not in the brain. And so, like if you have if you utilize a epidural block for anesthesia, like when a woman goes in for pregnancy and they give her a epidural block, a lot of times they're disrupting and utilizing these glycine pathways to stop transmission of any signal, any pain signal that would be going to the brain. Our other categories, so the other four, the peptides, purines, gases, and lipids, we're just going to kind of do some light lip service to these ones at this time. Our peptides, right, these are proteins. The great example of compounds found in this that are neurotransmitters are opioid peptides. So our enkephalins, our endorphins, we utilize these peptide compounds when we're making our analgesia drugs, our pain relief drugs, opioid drugs, right? Like OxyContin, those aspects. They're utilizing these pathways to inhibit neuron signaling. We have purines. Okay, purines are, again, a compound utilized often. Purines are in our DNA. The most common purine we utilize is adenosine and adenosine monophosphate, AMP, right, and adenosine triphosphate, ATP. So ATP should be familiar to you guys. That's the energy source in the body that we utilize for energy. AMP is when we've taken off two of those phosphates. But even though we use these for energy, they can also be used as a neurotransmitter as well. So some tissues have what we call purinergic receptors that can bind to ATP. We have an AMP. We have these in the heart and the central nervous system. An example of a neurotransmitter that's a gas, nitric oxide. Right? Nitric oxide, it doesn't have a membrane receptor. It doesn't bind anything, but being a gas, it can diffuse freely into the target cell, and it can then disrupt cellular processes and act as a neurotransmitter. And lastly, we have our lipids. The lipid neurotransmitters that have been studied, generally an example would follow what we could talk about is like our eicosanoids. These are the compounds like THC found in marijuana, we classify those as our cannabinoid compounds. These combine to receptors and again elicit a cellular response. So these kind of go through and outline all these. I don't expect you to know all of the stuff in this table. I put this up here. The ones that we will be talking about the most are going to be acetylcholine. I do expect you to recognize that they are receptors of acetylcholine are cholinergic, that we have nicotinic, muscarinic, where those two are primarily found. The agonist, again, agonist means a compound that also triggers a response. The agonist of nicotinic is going to be nicotine. The agonist of muscarinic is muscarine. The antagonist, so something that would stop that. On the nicotinic, we're not going to really talk about these too much, like bungarotoxin. But on the antagonist of a muscarinic is atropine. So you may have heard atropine before being administered to kind of as a pain, is it like an anesthetic when you're doing surgery? Sometimes they'll have an atropine drip going on the actual tissue itself that they're cutting into. That's because it blocks these receptors, so pain, they can't be, those tissues couldn't be sending signals. When heart muscle, sometimes we'll take atropine for. Okay, so that's a big one. Then the amines, norepinephrine, its receptors are adrenergic alpha and beta, and that's really the big one, but kind of recognizing that dopamine, histamine, serotonin, those also fall into the amines. Then the amino acids, the ones that I want you to know are GABA and glutamate. So glutamate being excitatory in the central nervous system, GABA being inhibitory.